Listo. Bueno, buenas tardes nuevamente y les doy la bienvenida a los participantes en línea. Y bueno, pues como saben, esta es la última clase del curso y vamos a hacer algunos cambios. Como se tiene que realizar el examen al final del curso, el profesor va a terminar media hora antes, es decir, va a terminar a las 3:30. Entonces, a las 3:30 se les va a enviar el examen a los asistentes en línea para que lo resuelvan y lo tienen que enviar al correo que se les va a indicar para ya evaluarlo. Entonces, el examen va a ser de 3:30 a 4 y se le está dando 15 minutos más para que lo completen. Entonces, a más tardar a las 4:15, ustedes, los que están en línea, envían el examen a los correos que se les va a indicar, que es a la doctora Neftalí Rojas y a mi correo. Y con eso daríamos por terminado el curso. Entonces, a los asistentes se les va a dar, como ya se los ha comentado, una constancia de asistencia. Bueno, pues para no quitarle más tiempo, les dejo la palabra al doctor Christian Abendroth.
Okay, thank you very much. Hi, everyone. Let's start here. So let's pick up where we stopped yesterday. I will just repeat a few slides. We have maybe 30 slides left. I don't know how much time I need for this, and we want to finish a bit earlier today. If we are really fast with this, I can start another lesson which goes a little bit about into the methods for proteomics. And if not, we can then just stop after this lecture. So we shortly repeated this electron transport chain for the light-dependent reaction in phototrophy. And we discussed a little bit about how phototrophy would be affected when we have no oxygen. And then we came to this figure. So here we asked the question, what is different to the scheme that we knew before from the second presentation about photosynthesis. And I think someone, I don't know who it was, but someone found out that there is actually just one electron involved and no water electrolysis, so we have actually no production of hydrogen, which is not so nice because the hydrogen would be really interesting to chemically reduce carbon dioxide in order to produce more methane. So basically we have one electron which circulates over and over again through the reaction center and the BC1 complex. So it's basically recharged in the reaction center, then it goes into the BC1 complex. Again, this is like a pump for protons, and then the protons are pumped into the cell in order to increase the concentration, to have a gradient of protons, which could then power the ATPase to produce ATP. So we were then talking about the question, what impact would we expect from light in anaerobic digesters? And well, one hope is that we could chemically reduce CO2, but accordingly to this picture, this would not be the case. Maybe there's another mechanism involved that we still don't know. And this would also be the basis to produce more biomass, to have more substrates that we can actually use for anaerobic digestion. But maybe even if this is not happening, the photosynthesis could help to improve the degradation efficiency due to a better oxidation potential. Maybe it could also cause microaerophilic conditions, but again, this would only happen if we would have this electrolysis of water. An interesting point is whether photosynthesis could help to increase the rate of direct interspecies electron transfer. Well, we talked a lot about this direct interspecies electron transfer, and we will come back to this question. We will partially answer it. There are some experiments on this which give a good hint whether this is possible or not, but the question is not finally resolved. And there's also the possibility that the methanogenic pathway is affected. This was discussed yesterday in the introduction, where you have seen that there is this coenzyme M bound to a methyl group, and this methyl group can be released if the coenzyme M reacts with coenzyme B. This is the final step of methanogenesis in the methanogenic archaea. And this reaction to release the methylic compound from the coenzyme M can be triggered by light, but this has only been shown in vitro. So they extracted actually this enzyme, and then they have shown this in reactions in the lab that this is working. But no one has shown this or has given final proof that this is also happening in the microbes themselves. But it could be a good possibility. Yeah, this release of the methyl group from the coenzyme M has actually only been shown for this enzyme, coenzyme from Methanothermobacter thermoautotrophicus. So this could also be a problem. So even if it works in vivo inside this organism, we don't know whether it works in other methanogens as well. So there is still some work ahead of us to find out whether this is true or not. I also told you that there was already a first patent, so we are not the only ones thinking about this option to apply light on anaerobic digestion processes. So people are working on this for a long time. So far it was not transferred into the industry, and I suppose that one reason for this is that it's really difficult to get a light inside. We will talk later on about one solution that we could have here, but it's not too easy. Well, and then we saw this biofilm that we got luckily in summer in Germany in our construction hall with a transparent reactor, and the hall was light flooded, and then suddenly this biofilm appeared, microbes turned red, and we sequenced this. We are not the first ones who are working with light in anaerobic digestion, but we are at least the first ones who sequenced such a biofilm. So there is another article that we will talk about a little bit, but this article was not talking about biofilms. Apparently they had the phototrophs inside the sludge. At least that's how I understand this article. They didn't prevent any photos. Well, what did we do here? So we had this reactor fed with sewage sludge and grass biomass. It was a two-stage system. And then when this color appeared, I asked people to retain the biomass. So they released the sludge, and then I got a chance to scratch the biomass from the inner surface of this reactor. And then I sent it to sequencing. It took approximately 3 weeks until this biofilm appeared. We applied 16S sequencing and also true metagenomics. We compared both, but I will not go into this. I will more focus on the metagenomic sequencing. So if you're interested in this, you can read the full article where you also get the information from the 16S data. And based on this shotgun metagenomic sequencing, we then performed a taxonomic analysis and a functional analysis, and we will have a look at it now. So those are the organisms that we found based on the true metagenomics. And it's already interesting because you know how diverse the microbiome is usually. And we were already here on a family level, so it's much more specific than the phylum level. But still, there were 3 major organisms. There are several which are just there in a small abundancy, but apparently we had just 3 organisms which were really abundant here. We did not only perform this metagenomic sequencing. We had a lot of sequences, and they were enough to also close the genomes. Maybe you remember that we talked about the true metagenomics, where we are kind of puzzling together each DNA fraction in order to create closed genomes. And with this, you can explain the capabilities of the microbes much better. You can even find new organisms, and we also find some. And with this, we were also able to classify those ones on a species level. But before we did it, we looked for potential phototrophic groups. And there are some of them. The most abundant one is Rhodopseudomonas faecalis. But then we also had these two minor groups, which are also phototrophic. Always when it starts with rhodo something, they're usually phototrophic. So we had quite some phototrophs inside this biofilm, which makes sense, of course, because we saw all these pigments. So now we created these genomes based on some bioinformatic works. And well, we find out that this one is the Rhodopseudomonas faecalis. And we found also that there was the Methanesaccina matsei. Maybe you remember the Methanesaccina. Where did we find it mostly? Do you remember? Raising? No. Okay. Anyone has an idea? Okay, so maybe you remember these 3 diagrams from the 7 biogas plants that we sequenced. And there was this leach bed system, and especially in the leach bed we found Methanesaccina. And we were really interested in Methanesaccina because it has several interesting capabilities. It's extremely robust. It's really diverse on the metabolic level. And then we found also this Fermentimonas canicola. I don't know what's this guy doing exactly. It's a hydrolytic organism and can also engage in acidogenesis. But there are many organisms that can do this. So I wonder whether there's any specific reason that we have especially this Fermentimonas canicola enriched in this phototrophic biofilm. So maybe it's also phototrophic, or it's somehow an acidogenic organism that can perform direct interspecies electron transform to engage in a strange way with Methanesaccina matsei and Rhodopseudomonas faecalis. So we don't know. Those are just hypotheses which we still have to follow. So here are the genomes that we could close. You see they're numbered from 1 to 16. But we didn't show all of them in the paper because some of them just hadn't enough coverage and the quality was not good enough. So we have just shown these ones. And you see that some of them are a new species. And there's even one of them which could be a new class or a new order. So you can look for the closest relative that you find here. But then if you would be able to culture them, you could give them a new name. So those are really new microorganisms that haven't been described before, which again highlights the novelty of this biofilm. So I think this makes it really interesting to further study it. Well, we then started to analyze the genes that we found in these genomes that we assembled from the metagenomic set of data. And we found 108 different glycosate hydrolases. So those are so-called carbohydrate active enzymes, enzymes which help to degrade sugars, all kind of polysaccharides. So basically we found important enzymes for the hydrolysis. If you remember the important steps of methanogenesis, so this is the first phase from the 4 phases of the methane formation from anaerobic digestion, followed by acidogenesis. Again, we found some genes that are involved in acidogenesis, and some of them were involved in lactate fermentation. And then we also found genes involved in propionate formation. So apparently, there are organisms that are taking up the products from the hydrolysis to form several acids, and this must be our Fermentimonas canicola, or maybe the Rhodopseudomonas fecalis? We don't know. I mean, we could study this in more detail, but money was restricted, and I would need more money to pay the bioinformaticians to dwell deeper into this. So that's what they delivered to me. Well, we also found some genes that are related to acetogenesis. So this was also interesting. Apparently, the organisms were able to take up the lactate and the propionate and to convert it into acetate. And then, no surprise, we also found genes responsible for methanogenesis. I was surprised that we only found genes for the heterogenotrophic methanogenesis. So there are differences if we go to the species level. There are some Methanosacina which can actually perform all pathways, and some are only able to perform the hydrogenotrophic pathway. So it seems that we found one which is only able to perform this hydrogenotrophic pathway. Nevertheless, we found all 4 phases. So this means that this phototrophic biofilm was able to perform all tasks that are required to perform biogas, to perform anaerobic digestion. Well, in the next step, we started to compare these results to things that have been described in literature. And there was an interesting article from 2018, and it was on Rhodopseudomonas palustris. We don't have Rhodopseudomonas palustris in our biofilm. We had Rhodopseudomonas fecalis, but they share a similarity of 99%, so they are almost the same. So it could be that we have similar capabilities in Rhodopseudomonas fecalis as in Rhodopseudomonas palustris. And here we see the electron transport chain. That's the one that we discussed earlier, where we have always the same electron circulating in the membrane in order to activate these pumps to create a gradient of protons that can be used for ATP production, which you see here. And then it was interesting that they are using actually the ATP to produce more biomass. In this case, they're able to fix nitrogen from the atmosphere. This would be probably not so interesting for our anaerobic digesters from agriculture because we're not producing any nitrogen there. But maybe this could be important for water treatment. So the electron source was here thiosulfate. But then there is another picture which shows also the possibility to use acetate as an electron donor. So it goes into the central metabolism, and from there electrons are retrieved and they can be used together with the energy from here to produce biomass. Well, what does this say us? So it does not support the idea that we are producing more hydrogen from electrolysis of water, but still it creates some additional energy, and maybe this can create some more oxidative force to power processes that we need to better degrade material, for example, to engage with the electron transfer, with the direct interspecies electron transfer. Okay, here again we see these 2 graphs from well, originally there were 3, but I just showed 2 because now we're talking about high performance reactors. And then we see these 2 organisms which are most abundant. Oh, sorry, this is wrong. This should be Methanoculeus, the blue one. And then we have here Methanosacina. So apparently Methanosacina can be abundant in the high performance plants. But sometimes we have Methanoculeus and we don't know why. We don't know under which circumstances and which selection pressures Methanosacina is able to suppress Methanoculeus or the other way around. We would like to find out. I'm looking for a while in the literature. I also tried to do some own experiments on this, but so far we didn't find anything interesting in this regard. There are some hints. For example, we know that metanosatsina is very robust, can resist to high amounts of ammonium. It survives high contents of salt. Also, a high chemical oxygen demand can be applied. And also, it can survive relatively low levels of pH. This might help to better select metanosatsina, but it would probably also help to enrich metanoculeus. So we need some new selection pressures here. One selection pressure could be viscosity. So there's one hint in this diagram here. Maybe you remember that in the leach bed system, we have in the leachate, which is trickling over the pile of biomass, a very low amount of total solids. So it's basically liquid and we can pump it easily. And then we get an increased amount of metanosatsina. And I was also performing one article where I used sewage sludge, which usually contains metanotrix, or how's it called, metanocyta. So it was renamed recently in metanotrix. And when we then increased the loading weight with a biomass which is high in solids, the viscosity increased continuously. And then at some point, it was taken over no, it was not taken over by metanoculeus, but the control reaction that was fed only with liquid substrates suddenly was increased in metanosatsina. So this was a really nice hint that we actually can increase metanosatsina when we reduce the viscosity. But it's just one idea, and another idea could be the light. So it's also metabolically very diverse, so it can eat directly acetate, as I have shown you. Unfortunately, not in our biofilm, but there are metanosatsina genes, species which are able to do this. They can grow hydrogenotrophically and also acetoclastically. So we might also add actively acetate to our reactors in order to improve the growth rate for metanosatsina. And then we have the direct interspecies electron transfer. We have also seen that metanoculeus, or I at least talked about it, that metanoculeus is not able to perform this direct interspecies electron transfer, but metanosatsina is. So in the moment when we are providing conductive particles, maybe this improves the selection pressure for the metanosatsina. Well, here again the direct interspecies electron transfer. I guess you still remember it. So here we have the mediated interspecies electron transfer, where we have like organic components carrying the electrons that we want to transport. And then we have the direct transfer of electrons via nanowires between the organisms, or we have conductive particles that we throw actively into the sludge, like magnetite or activated granular char. There was an interesting article in 2021, and there it was shown that actually Rhodopseudomonas palustris was able to perform a direct interspecies electron transfer. And this experiment was done in this kind of energy cell with a cathode and an anode. And at the anode you had Geobacter metalloreducens, and then at the cathode we had Rhodopseudomonas palustris. And then after a while they did it under dark conditions. I don't know why they did it under dark conditions, but under dark conditions they saw that eventually the voltage was increasing. So it seems that there was an electron transfer from this organism to this one. So how is this now important for our biofilm? Well, we could imagine that we actually have a similar system with metanosatsina. In this case, the Rhodopseudomonas would have to be here at the anode, and we would have to place metanosatsina here to the cathode, because methanosacina would be the final electron recipient in order to use the electrons for the chemical reduction of CO2 for the production of methane. Well, actually, just last week I saw an article. I have not been able to implement it yet in my lecture, but there was a new article from 2022, and they have done exactly this. So now they have proven that there is really an electron transfer between methanosacina and Rhodopseudomonas. And this is really nice because now it seems more likely that there is a direct interspecies electron transfer going on in our biofilm. And this would mean that Rhodopseudomonas is able to interact with light and at the same time to send electrons to methanosacina, which means that there is somehow an influence from the light onto the methanogenesis. Well, we compared this further. I told you that there was another article which was working with light. They didn't say anything about a biofilm. They talked about there were several reactor setups that they used, and they were using some light to illuminate these reactors. I gave some conditions here. I'm not going too much into the detail of the construction. Those are basically also transparent reactor where they applied some light. And in one case with 500 milliliter reactors under thermophilic conditions, they saw after 8 days 3 times higher efficiency in the methane production. So seemingly there was an impact. There is another setup in the same article, and here they used much less sludge. Was 15 milliliters medium with 5 milliliter sludge. And then they were using hydrogen as a substrate and compared it to acetate. And apparently it works best when they used hydrogen as a substrate under these phototrophic conditions. So this is interesting because this fits very well with the things that we have described in our article, where we have seen that we found mainly hydrogenotrophic genes in the methanogens that we found in the biofilm. So this fits very well. They did a third experiment, and here they used a more specific light wave. They used blue light. I don't know why. They didn't explain it too much in the detail, but then they saw an increase of methanosacina, which again fits very well with the things that we have seen in our biofilm. So with this we have already 2 articles supporting the idea that we can have this light-active biofilm where all phases of methanogenesis are supported and where light can somehow facilitate methane formation. Well, and then it could still be possible that there's another interaction with light and methanogens, a more direct interaction where light is actually activating the methyl coenzyme M reductase in order to release the methylic group from the coenzyme M, which might fasten up the formation of methanogenesis. Well, there's another article, and they didn't go into the metagenomics so much, but they had a really interesting setup. And those were the Sawayama et al. From 1999. Those are the same guys that actually patented this process. And they used a so-called LUASP. This stands for Light Upflow Anaerobic Sludge Blanket. Do you still remember what the UASB reactor was? Yes? You are nodding? So what was it?
It was the same, but without the light. The all-flow anaerobic sludge blanket, when the reactor have a blanket of sludge like in the middle. So
yeah, that's right. Why?
And the water comes from above to, no, from bottom to
yes,
up.
That's also right. And What's the selection pressure there? Why can we use this to separate actually liquids from solids? Why does this work in this kind of reactor but not in our continuous stir tank reactor? Yeah, you have an idea?
Because of the granules that are the microorganisms form, so they can precipitate inside the reactor to the bottom, so they are not washed out.
And why do these granules form?
Because of the condition of stress inside the reactor, they produce extracellular polymeric substance, so they can act as a glue.
Yes.
And they form the granule.
But what is the stress? Why are they doing this?
I think because of the
upflow.
The outflow, yeah.
That's right. So it's totally right. So all the microbes which are not forming granules, they will just flow up and they will leave the reactor, and then only the ones that are able to grow in granules are retained. So this is our selection pressure. And this means that we have this blanket, and then we have regions above where the sludge is much less dense. And this facilitates the light inflow. So therefore we could use this combination of an anaerobic upstream anaerobic sludge blanket in combination with light, with illumination of these reactors. Okay, so with this we are basically done with this part of the lecture. So we have seen that we have all phases of anaerobic digestion taking part in our phototrophic biofilm. We didn't find any cyanobacteria. We found basically purple bacteria, which is a shame, because with the cyanobacteria we would have actually the electrolysis of water and also maybe some production of oxygen to cause microaerophilic conditions, which could improve the degradation rate in such reactors. And amongst the phototrophic microorganisms, we found especially Rhodopseudomonas, which is involved in CO2 fixation, and maybe it can also support the methane release. We found a very high abundance of metanosatsina, which raises the question of whether there is some direct interspecies electron transfer involved between Rhodopseudomonas and metanosatsina. Recent articles indicate that this might well be possible. And then we have no clue, at least not yet, what's going on with this Fermentimonas canicola. So maybe it has some hidden superpower that it can combine hydrolysis or acidogenesis somehow with light, or also engaging with direct interspecies electron transfer with Rhodopseudomonas or metanosatsina. I hope so. I could imagine, because there is no other, I don't see any other explanation why only Fermentimonas canicola is enriched when we have so many different hydrolytic and acidogenic bacteria that could be involved here. Well, that's it. So what does the time say? We have still half an hour, so we can start the next lecture. Can you please change the presentation? Okay, thank you very much. So this is chapter number 6. We just finished chapter number 7. I told you yesterday that I wanted to be able to finish chapter 7 because this was most important for me, and it also gave us the possibility to come back to the photosynthesis that we already discussed so intensely. And well, now we still have some time to at least sneak in a bit into chapter 6. Let's say how far we are coming. This is now about proteomics. And this means we're talking about how to analyze proteins and how to perform a proteome analysis and anaerobic digestion. We already got an insight into this. You remember that in our chapter number 5, I presented you one article where I combined metagenomics with proteomics. And here I have another article. I don't know whether we come to this because I started with some methodological aspects on how to analyze these proteins. And for this, we are actually using mass spectroscopy. Do you all know this? I could imagine that it's also important for engineering to analyze some materials. I don't know. Have you ever heard about it? Mass spectroscopy? No? Yes? Yes? So for you it's not completely new. The other ones? No, no? You? No? No? Okay. Yeah, so it's a technology which is applied very often, and it's not just applied for proteomics, but it's usable for proteomics. And the idea is that we have here this part where we have the entry of some substances, and we try to disrupt them into smaller molecules and to ionize them. So we have here an electron trap which helps you, some kind of charged element, which helps you to retain charged particles. And then they are accelerated through this little opening into a magnet. And then, depending on how heavy they are, depending on the ratio between mass and their charge, they take a different course in their acceleration. And then you can detect them. You can see where you can detect them. And with this detection, you can actually measure the mass to charge ratio. Well, and these values are very specific. And with these values, you can then identify your substances. There are different concepts. This is a very simple one. There are more novel systems that are applied. Here's one video, which is really nice. So can you please show the video?
The Abendroth process.
So there's no sound.
The Abendroth process is a two-phase procedure. Initially, the samples are fixed in a crystalline matrix. In the initial ionization step, they're bombarded by a laser. The sample molecules vaporize into the vacuum while being ionized at the same time. High voltage is then applied to accelerate the charged particles. The second step is the time-of-flight mass spectrometry phase. In the linear mode, particles will impinge upon the linear detector within a few nanoseconds after ionization. Higher mass molecules will arrive later than lighter ones. Flight time measurements make it possible to determine molecular masses directly. Each peak in the spectrum corresponds to the specific mass of the particle along the time axis, starting with the ionization moment. In the reflector mode, the particles are diverted so that they fly towards a second detector. In addition to extending the flight distance, the reflector also focuses the masses. The combination of these two effects makes for higher resolution than in the linear node.
Well, it is a really similar concept as the one that we have just seen. just with the difference that we are not measuring the way they are going, but that we are measuring the distance, how long they are flying or how fast they are flying across a certain distance. And then there is another concept which is applied nowadays really often, a so-called orbitrap. This is an orbitrap system, and here you have your electron, your ion trap again. So you're conducting your ions into this room, which is crossed by this spindle, electrode spindle. So it's basically an electrode. And then the ionized particles are circulating around this spindle. And since this is an electron, you can measure this, and then you get a signal like this one. Well, and then depending which ion you have, you will have a different pattern, which you can analyze. And very often this is repeated over several steps. So you have your sample, you have your ionization with some smaller fragments. You separate them. Then you can analyze, no, sorry, here you already analyzed them the first time. And then you separate them again, and you can try to make them collide with other particles like non-charged gases, for example, to be sure that the colliding substances are not giving a signal themselves. So they are getting split into smaller molecules, and then you can measure them again. So then you know basically the mass to charge ratio of bigger particles and the mass to charge ratio of the smaller particles resulting from these larger particles. Well, how do you analyze this? In the end, you get a spectrum like this one. And you have to know how this spectrum should look like in order to know what you are seeing there. So someone must have tried this with exactly the same substance before. This is then fed into a database. And then once you analyze something unknown, you can see whether you find your spectrum in this database or the certain peaks. And then you can analyze what you have there. Well, this is a really simple scheme. It's just with one substance. You can imagine how complicated this must look like if you have hundreds or thousands of different proteins from an anaerobic digester sludge. So this goes really deep into informatics. I have no clue about how to do this, but for me it was important just to generally understand what they're doing here and how it works, because I work with this taxonomic profiles from proteomics, so it's nice to know how they are actually created. Another thing which makes this very difficult is that the fragments are not always the same. So if you have this molecule, for example, you can have, after your ionization, several molecules which have, for example, a break here at the alpha bondage or a break here at the beta bondage. So this is counted based on the heteroatom. So you have your molecule, one heteroatom, which has usually a higher electron pulling force than the other ones, the other molecules, other atoms there. And then counting from here, you have alpha, beta, gamma, delta, epsilon atoms. And then the bindings between them are also called with alpha, beta, gamma, delta bondage. Well, and this is one example how you can have this alpha cleavage. So here you have a substance, and then after ionization, here is a positive charge, but usually this oxygen is quite electron pulling, so it will try to get its electrons back from somewhere. So it pulls them out of the molecule, and then this causes a disruption, a cleavage at this position, and in the end you have these two molecules. So this is a really simple, molecule. Imagine again how complicated this will be if you have really large molecules, proteins, where you have hundreds or thousands of amino acids. Then it's it's a large amount of fragments, of different fragments that you have to deal with. And then, as I told you, there's not just this alpha cleavage, but there could also be a beta cleavage, for example. The principle is the same. So you have here an ionized oxygen, it tries to pull the electrons back from somewhere. Here it has some access to this flexible tail, and it pulls electrons from this hydrogen atom. Again, this causes a disruption, and then you have this molecule and this one is also how is it called? It's like an isomer. It's isomerizing into different forms of the same molecule. Well, and then as I told you, you can create this spectrum to see what you actually detected. So how are you doing this with proteins? How do you start such an experiment? Because first you need your protein fragments somehow. So you start with some cells, some tissue, or our sludge. You have to extract all the proteins. There are chemical kits that you can use. Then you have, like, I don't know, 15, 20 reaction steps that you just follow. And then based on chemical characteristics, you are able to retrieve the proteins from the remaining cell fraction. And then you put it on a gel, on an electrophoresis gel. I don't know whether you have seen an electrophoresis gel before. I have another picture later on. So it's basically a gel, and you are pipetting your proteins onto this gel, and then you have some highly conductive liquid, and then you apply some electrodes, one before and one after the gel. And then the liquid is kind of pressed or pulled through this gel, and it's carrying the proteins with it. And then depending how large the proteins is, they will go better or worse through the gel, forming these kind of bands. And then you can actually cut out one band with a particular size, and then you can digest them. You can use protease, enzymes that are destroying proteins, and like this you get already smaller fractions. Now you could bring this all into a column of chromatography. So this means you have again a matrix similar in the gel, and your fragments are sorted according to the size again. And then you're ionizing them in order to perform the mass spectroscopy. You have your first stage, then you can make the particles smaller again by collision with a neutral gas, and then you have a second detection, and then you have these spectra. Again, this is a very simple spectrum, and well, if you have a lot of different proteins, it gets more and more complicated. Oh, now I explained already everything. I wanted to go through it step by step, but I guess you could follow me. Right? Are there any questions? All okay? Wait. Here's a database that you can use for this. It's just an example. Mass Bank. I was just playing around with it a bit. So you could go on search. It's a free-to-use database, so you can just enter the webpage and the link into the webpage, and then you get it. And then you get like a search line similar to Google, and you could put in here, for example, the mass 92. You can put whatever mass you want. I just used 92. And then you get already some results. And then I was just checking one of them, and then I got a lot of information about this one substance. So this is how it works, but I mean, this is quite time-consuming and manually, so there must be some way to automate this. So this again goes more into informatics and bioinformatics, and is out of the range of my own expertise. Yeah, if I take this substance, I can also check the spectrum here, and this would look, for example, like this. So I can directly compare the spectrum to the one that I measured. Well, with this I would like to summarize this. So we have seen that mass spectroscopy is one of the key technologies to analyze proteins, and we use it for protein sequencing, although mass spectroscopy is also used for other things, not just for proteomics. Usually we measure this ratio of charge to mass, and it becomes complicated due to the structural changes. We have these cleavages, the alpha and beta cleavage, which can already create a diverse spectrum for a simple substance, and it becomes more and more complex the bigger the molecules are and the more complex our matrix is that we are analyzing. Well, to analyze this, we have to separate our proteins. We usually do this with this sodium dodecyl sulfate gel electrophoresis. I will show you a picture again to make it a bit more clearer. And then we can have multiple mass spectroscopy steps in order to observe our molecules at different resolutions to have it more accurate in the end. And the final result has to be compared to some kind of database, which is kind of similar to the analysis of the 16S data that we already talked about. There we also create our sequences, which we have to compare to some kind of database in the end. Kind of 16S was green genes and silver. Okay, so I hope we still have time. 20 minutes, so we can start this article here. Well, this is now one article which was really focused on metaproteome analysis. It was published from a colleague of mine. I think it's a really nice work. They started with a very comprehensive table of many biogas plants which they investigated. It's very difficult to see anything here because there's so much data. But one interesting thing that I observed here is, for example, that the thermophilic plants are much better in the organic loading rate and also in the biogas production. That's something that we heard before already, that thermophilic reactors, once they adapted after a long time of the adaptation, can become very efficient. Yeah, I think the other parameters, they don't matter so much for you. They might be good to know if you want to make some statistical analysis to compare them to the proteomic data that they produced, or in order to have a complete picture of all the biogas plants that were analyzed here. But I will not go much into the details of this. So the aim was now to extract from all these biogas plants the proteins and to analyze them. So how did they do it? So they used basically the same method that we know already. They started with this SDS page, followed by chromatography. Then they had the Orbitrap analyzer, and then they compared it to a database. This is how such a gel looks like. So first we have our proteins, and usually we mix them with a substance called SDS, which stands for sodium dodecyl sulfate. And this substance has the capability to bind at all kinds of amino acids. It will dissolve all bondings between different protein fractions, and it will kind of linearize them. So in the end, you have not a globular big protein anymore, but you have smaller fractions which are linearized, and then you put them on your gel. And it's really jelly-like. There's a substance called polyacrylamide. You heat it up a little bit, and then you pour it between these two glass walls. Then you wait a while until it polymerizes. And then you have this jelly-like gel between these two glass walls, and you have some pockets on top of it. And into these pockets, you can now pipette your linearized, denatured proteins. Sorry, I'm going too fast. So we are now here. So we are now pipetting our proteins into these jelly-like structure, these jelly-like pockets. And now we apply a buffer with some electrodes. So you have one buffer on top of it and one buffer below. Then you have anode and cathode. And you have a really conductive liquid which is now running through your gel, carrying all the proteins with it, so that the proteins are separated by size. So the big proteins are just not going so well through the gel. They will be retained behind, and you first get the small proteins, and then you cut them out. You get, this is how the gel finally looks. And then you can take a scalpel and you just cut out the small fraction. Usually you have a marker here, and this marker tells you certainly sizes. So usually you know exactly which size this is, which makes it again easier later on to interpret your data with the mass spectroscopy. Well, then they take these proteins which they cut out and they put them into the mobile phase, some liquid phase, liquid buffer, which they are pumping into this chromatographic column. This column is filled with some material. Very often they use saccharose. And then again, the bigger fractions, the bigger molecules are going not so well through the saccharose, so you will get the small particles. First, you can detect them based on optical methods, and then you can start to perform the mass spectroscopy. And here they used this Orbitrap concept. Well, in the end, they used a database. They built actually their own database based on SQL and several processing stages. And their database was able to interact automatically with the UniProt database. And in the end, all these calculation steps or treatment steps were transferred into one Excel file where they had their classifications to work with them. So you see it's already some informatic knowledge required to perform this. And I used this data to produce, for example, this dendrogram. I don't know whether you have seen something like this before, but it basically shows the distance of the different proteomes. So they take the entity of the whole proteome from one sludge, and then they align it similar to our cluster alignment that we used with the DNA to compare the similarity of different DNA strains. They do the same here. And then the closer you are to the right side, the more similar they are. And the closer you are to the left side, the more different they are. For example, these two samples are very different, are very similar. But these two samples are very different from these two samples. Is this clear? Yeah? Okay. Yeah, they also made some information, or gave some information, how they clustered, for example, in the top part, you found the laboratory samples, so they had not only industrial biogas plants sampled, they also sampled some biogas plants from the lab, and it was interesting to see that actually a reactor in the lab behaves totally different from a reactor in industry. So this puts actually in question how trustful our results from the lab are. So this is quite interesting. And then you have here one big cluster for the thermophilic reactors and for the mesophilic reactors. Yeah, you can also see some color which highlights similar clusters. Here they have some numbering for the different plants they analyzed and the time points. So you see they had always duplicates. And the duplicates are always the samples which are closest to each other, which makes sense, of course. So now they wanted to do something more with it, and they produced this so-called Corona plot. This is actually quite nice. So if you have such a Corona plot, you can click on it, and dynamically it expands and becomes smaller again. So you can say that you want to expand maybe just the archaea, and then all this becomes one pie diagram. So it's a really dynamic way of showing data. And you can download this as a macro. So it's really nice, and it's not difficult to use. But here in the paper, they just used the perspective where you can basically see everything. We saw this already yesterday when we talked about viruses and bacteriophages, and this came actually from this article. And you see most of the proteins are related to bacteria and archaea. Well, then we have a lot of Firmicutes, Proteobacteria, Actinobacteria, Spirochetes, and Eukaryota. And this is basically the same what we know already from our taxonomic studies based on sequencing. So this fits very well together. Just for this, we don't have to perform proteomics. Then we have several unassigned organisms where we just don't know what it is. And then we have the viruses and the bacteriophages. This is now something interesting. This is something which justifies the application of proteomics in this case. What do you think? Why should I not be able to see bacteriophages and viruses based on metagenomics or 16S sequencing? Imagine I repeat the same study, but just on a DNA level. Would you expect to see the viruses here and the bacteriophages? You're nodding? Why? No idea? No. Maybe here, some ideas.
Can you repeat the question, please?
Yes. Why would I do proteomics in order to analyze viruses? Why not doing the same based on metagenomics or 16S? Imagine that I repeat the whole procedure, what they have done, but instead of proteins, I would analyze the DNA. Would you expect here something else, something different? You have a, yeah?
I think that's because viruses don't have proteins on their own. They need someone else to put the DNA, and then the machine of the thing that they are infected will produce that protein. Mm-hmm. So viruses by themselves don't have proteins, so you can analyze.
Goes into the right direction, but they have proteins. They are actually built out of proteins. The entire virus is a protein. But they don't always have DNA. Sometimes you have viruses which have DNA, and sometimes you have viruses which have just RNA. So you might be able to detect a small fraction of the viruses, the bacteriophages. But you see that this is already quite small. So if you have even less, then it might be difficult to still see it in your taxonomic profile. So with proteomics, you actually have the possibility to analyze the totality of all viruses and bacteriophages involved in the processes. I'm always saying both because a virus usually attacks humans. Bacteriophages are attacking bacteria, but then we have also the methanogenic archaea. And when something is attacking the methanogenic archaea, then it's a virus again. So that's why we use both viruses and bacteriophages. And then we have this, which is also something specific for proteomics and more difficult to achieve on a DNA level. We could have a similar map on DNA level with metagenomics, but this would then just represent the capabilities of the involved microbes. And with proteomics, we really see what they have done. So this is information which has already been transcribed and then translated into proteins. So that's actually not what they are doing right now, but what they were doing just before. And this is something you can just do with proteomics. And to give you some idea how to read it, it's all very small, but I indicated some interesting things with arrows, and I was writing it to the left to highlight what you should see here. For example, the blue arrow indicates a big group of microorganisms, the Bacilliaceae, and the green one represents the rhizobioaceae. And they are all connecting with a big line to the sugar transport. So all of them are involved in sugar transport. Here we have multiple arrows which are indicating activities related to cell structure. So they have to reproduce, they have to live. So they need to produce or to perform protein synthesis. They have to perform the cell cycle to divide, to produce more cells. They have to perform amino acid synthesis and transcription. Everything they need to exist. Here we have a green arrow which represents the Anaerobic, which is responsible for CO2 fixation. This was interesting. Why is CO2 fixation here involved? So maybe those are these kind of homoacetogens which are able to convert carbon dioxide in hydrogen into acetate. Could be. I didn't check on this organism before. Yeah, here we have our methanogenic archaea, and we have a big activity in regard to methanogenesis, of course, which makes sense, but also a very big activity to electron transport. And this again highlights how important electron transport is in order to understand and improve anaerobic digestion processes. This small field that you are seeing here, this is actually everything we have on cellulases. And this is really surprising because we have here agricultural biogas plants which are fed with biomass, and the biomass from plants contains a lot of cellulase. So wouldn't we expect to have more cellulases here? It's a question that I heard several times before. There's no sufficient answer for now, but for some reason we find little cellulases, although they're able to degrade a lot of the cellulase, at least with some time. And then that's also interesting, we talked already about how complicated the communication between those microbes is, how complex it can be. And well, we have here several arrows which are indicating microbial stress behavior, sporulation, stress response, DNA repair, and detoxification. So this is all related to this kind of communication between these organisms. All these results, I didn't present all of it. I highlighted some things that I found to be really important. And all this has to be taken with a grain of salt because there's always a bias with the database, of course. So it's always limited by your database. And you have seen in the assignments in the beginning that there's a large fraction which can't actually be assigned because the database just don't know what it is. There can also be some misassignments. So there can always be errors that you should take into account. Well, and then the viruses. What about the viruses? They are not represented here. I don't know why. But then they produced this really nice idea about how viruses could actually help to improve the process of methanation. And we talked about it yesterday already. So we have these prototrophic microorganisms and these auxotrophic microorganisms. The auxotrophic microorganisms, they can't grow very, very well. They have really specific requirements. But then the prototrophic microorganisms, they grow almost always and they don't need so much to be happy. But then the bacteriophages and viruses, they are destroying both kind of cells. And when the prototroph organisms are destroyed, they release supplements, nutrients, which could be important for the auxotrophic microorganisms to grow, which would be a big driver for biodiversity in our biogas plants. Well, with this we are done. We have seen that bacteriophages are really important for the biogas process, although little research is done on this field. I know maybe 3 or 4 works on this, so it's really underrepresented in research yet, but I think this will change in the future. And one problem is that we have RNA and DNA-based phages, so it's really difficult to detect them just based on DNA. That's why the authors here applied proteomics, and with this you can detect all of them. Well, we talked then about the possibility to show proteins based on omics approaches, in this specific case based on mass spectroscopy. We saw that the sugar metabolism is quite dominant in the overall activity in our proteomes, in the shared proteome of all biogas plants. Actually, we have seen that there is an extensive stress behavior, so it seems that the organisms are continuously fighting against each other. We also have seen that there's a substantial effort to grow. And we got an idea how bacteriophages are actually involved into the overall microbiome, and that they could have an important role in order to drive diversity by promoting the growth of so-called auxotrophic microorganisms. Well, with this we are done, and we exactly made it on the minute. So this is nice. Thank you a lot for listening. I know it was not an obligatory course, so it was really nice that you were coming here to gain some extra knowledge. If you have some questions, you can still ask them. Otherwise, we will proceed now with the exam. Yeah, it was quite short time to prepare for the exam. I hope you still manage. I tried to make the questions not too difficult. So let's see how it works. Thank you a lot.
Bueno, pues hemos terminado este curso que yo espero que haya sido de su interés y que esto también sea un inicio para que les abra las puertas a otros conocimientos. Marina, mejor, Reina, ¿me puedes ayudar a repartir? Son 3 hojas del examen para que Marina les mande por correo el examen a los que están tomando el curso en línea. Bueno, como les comenté al principio, a los que están presentes y a los que están en línea, vamos a tener un tiempo de entrega a las 4:15. 4:15. Entonces, por favor, lo mandan. Ahorita la maestra Marina les va a dar el correo de la doctora Neftalí Rojas y el mío, para que nos manden, los que están en línea, nos manden su examen a los 2 correos. Y los demás, pues nos lo entregan al final. También les recuerdo que preferentemente, y esto también para que ustedes practiquen, pues respondan en inglés, pero si alguien por más rapidez lo quiere contestar en español, está bien. Esto también para los alumnos, bueno, los asistentes en línea. Entonces, Marina, les mandas también a los que están en línea el examen y los correos de la doctora Neftalí y el mío para que lo regresen alrededor de las 4:15. Bueno, vean si hay alguna duda o alguna pregunta acerca de algunas preguntas del examen, pues de una vez lo vemos para que lo aclare el profesor. Bueno, pues adelante.
Sorry, one question. Okay, here. Yes. What is timber? Wood. Okay. Thank.
¿Alguna otra pregunta o duda? Para los que están en línea, si tienen alguna pregunta o duda, lo pueden mandar por vía chat. Luego, aquí afuera, por favor, de parar los árboles, compadre, Bueno, pues llegamos al final de este curso. Yo espero que les haya sido de provecho y que pues este sea el inicio de otras actividades académicas que vamos a tener. Como les decía, va a haber un curso de verano en junio, entonces los que estén interesados nos dicen para mandarles la liga para que se puedan inscribir. Bueno, pues le agradecemos al doctor Cristian, que nos acompañó en este curso. Yo espero que no sea el último, que nos acompañe en otros cursos más, y pues se le va a entregar un pequeño reconocimiento por parte de del SAC ambiental.
Muchas gracias, maestro. Muchas gracias, profesora. A nombre de la doctora María Neftalí Rojas Valencia, que es la presidenta del Subcomité Académico por Campo de Conocimiento en Ingeniería Ambiental, le damos nuestro más profundo agradecimiento por haber impartido este curso, que estoy segura que fue de gran utilidad para todos ustedes. Obviamente al maestro Constantino por hacer posible que esto pasara con todas las gestiones que hizo. Y bueno, pues agradecer a tanta gente, ¿no?, que está tras bambalinas, todo el personal de cómputo, personal del instituto que nos apoyó para que también esto pues llegara a buen término. Por supuesto a todos ustedes que nos acompañaron durante estas casi 2 semanas, tanto a los que están aquí presentes vía remota, los que están presentes aquí en forma presencial, como los que nos acompañan vía remota. A Marina también por haber llevado el control de la asistencia. Y bueno, profesor, en nombre del instituto, del posgrado, le damos este reconocimiento como una forma de agradecer el curso. Y un pequeño detalle para que se acuerde de nosotros. Gracias. Y bueno, aquí en México siempre va a tener las puertas abiertas en el instituto, en la UNAM, y que sea el primero de muchos cursos. Muchas gracias a todos. Gracias, maestro.
Bueno, pues gracias. La ingeniera Cristina preparó un pequeño convivio aquí afuera, entonces los que quieran y tengan tiempo, pues nos acompañan. Okay.
Thank you very much. It was a pleasure to meet you all, and yeah, maybe no, in Spanish not. So, German, ja. So, vielen Dank. War schön, euch alle zu unterrichten, und vielleicht sieht man sich mal wieder. Also alles Gute für die Zukunft und vielen Dank, dass Sie den Kurs besucht haben. Danke.